I'd like to do now is hand over to Dr. Kang Huang. Dr. Huang is a lecturer in the School of Chemical Sciences at the University of Auckland, and his research is focused on a combination of interdisciplinary approaches encompassing food processing, mathematical modelling, material science, microbiology, and molecular image to improve safety, quality, and sustainability of agriculture and food systems. So welcome to the seminar, Dr. Huang. And he will be talking about bio-based management options for sanitizing and disinfecting foods and surfaces. So over to you, Dr. Huang.
Thank you, Sue. Thank you for the introduction. Good afternoon, everyone. So my name is Kang, and today I'm going to talk about some food safety management approaches, which have been studied in our lab recently. So I don't want to spend too much time on the risk of food safety issues because I think David has talked a lot about the risk of cross-contaminations, but I just want to emphasize that the fresh produce and like the vegetables and the fruit product have the higher risk of cross-contamination than any other food categories. And also, every year we waste more fruits and vegetables as compared to other categories of the food product. So that's why we are looking at to reduce the microbial contamination of fresh produce because they have more issues than the other food categories. So during the post-harvest processing, as well as the transportation, storage, or retail levels, we need some good approach, which is green and clean labeling, and using low-cost materials, and also they are effective at low doses, then that would be more helpful because we can minimize the impact of quality attributes on the food. So in our lab, we are looking at the novel nature-inspired intervention approaches. Today, I'm going to introduce two of these approaches studied in our lab. The first one is the daylight-induced antimicrobial compounds, and the second one is the design of cell-based antimicrobial delivery systems. So first, the concept is that we are using the sublethal level of physical processes. In this case, we use daylight, and also we combine this low-intensity treatment with the low concentration of natural food grade compounds. And then the idea is that if we just use the low concentration of natural compounds, we didn't kill any bacteria. And if we only use the daylight, we don't kill the bacteria, of course. But if we combine them together, then we can get a significant microbial reduction in a very short period of time. So this is a rapid antimicrobial approach, and because we are using the low level of physical process, and also we use the low concentration of natural food grade compounds, so we can minimize the impact of the process on the food quality. So here is the one typical example, and it is widely studied in recent years. They are using curcumin, and also a very low concentration of curcumin, like 10 microgram per milliliter of curcumin, and combined with the UVA light. So not only UVA light, they can also use the LED blue light, the cool white light, even the sunlight. So if the light alone will not kill any bacteria, but if combined with 10 microgram per milliliter of curcumin, then we can get the significant log reduction in within 10 minutes treatment. So again, this approach can work at room temperature as well as the refrigerated temperature, 4 degrees C. And this approach works for different pH values as long as we have the acidic environment. So that means this synergistic antimicrobial treatment works very well for different light sources, the temperature, and the pH values. So one potential application is that we know in the grocery store, there's spray systems just assembled on the shelf, veg shelf. So we can spray this kind of low concentration of curcumin on the fresh produce surface. Then we turn on the light, and then we can get significant microbial inactivation, either sporage bacteria or pathogenic bacteria. So we tried the test on the spinach, lettuce, and the tomato, and we can get 2 to 4 log reduction on the fresh produce surface. So remember, we talked about the curcumin. We need acidic environments. That means it works only at low pH values, which is a constraint for the food quality. So that's why we want to explore more other candidate compounds which exert the same antimicrobial functions when they are irradiated by light. So of course, we can use we can try different compounds one by one, but we know it's not efficient, and also we know experiments take consume a lot of plastics, so it will generate a lot of greenhouse gas. So that's why we want to develop some simple approach. So in this case, we developed a high throughput and a rapid screening approach. So when we are looking at the mechanism of daylight-induced antimicrobial compounds, so we are looking at this simplified Jablonski diagram. So if we have a molecule and when they absorb the energy, they can transfer from their ground state, the S0, to their excited state, like the S1 and the Sn. And if they get to the Sn, which is a higher excited state, they will quickly return to their lowest excited state. And once they reach to the lowest excited state, they have 3 pathways. They can further transfer their energy via fluorescence emission, and also by the internal conversion. And the third pathway is the inter-system crossing. So if we are looking at the light-induced antimicrobial reaction, only this inter-system crossing is associated with the photoreactions. So in the presence of light with sufficient quantum energy, the molecules can transfer from the singlet state to the triplet state. Triplet state is a more stable state, which the molecules are preferred. So then they have 2 pathways can generate the reactive oxygen species. They have the type 1 reaction. We usually call it electron transfer pathway, and they have the type 2 pathways. So both type 1 and type 2 pathways, they can generate the reactive oxygen species. But the problem is we need a high efficiency of inter-system crossing. That means if we have the high efficiency of inter-system crossing, then we can generate more reactive oxygen species, which means that we have a higher antimicrobial property. So in this case, as we said, we developed a high throughput rapid screen approach based on the quantum chemical calculations, and also combined with the reactive oxygen species quantification approach. So in this case, the computational tool can help us to calculate the inter-system crossing efficiency of different compounds. And then once we identify the candidate compounds, we can use the ROS quantification approach to get to measure the exact reactive oxygen species generated by these compounds with some specific light sources. So in this case, we can identify the light sources. So here is one example of our most recent study. So we use the rose bangle as a reference compound, which is a well-known photosensitizer, which can generate a high amount of reactive oxygen species if they irradiate by the UVA light. Then we have two candidate compounds, and we found that these two candidate compounds will generate more reactive oxygen species compared to the rose bangle. And also we have a very good linear relationship between the ROS production and the irradiation time. So the x-axis is the irradiation time. So there's a very good linear relationship. So after we identify these two good, excellent candidate compounds, we tested their antimicrobial activity. So in this case, we combined the very low concentration of these two compounds, which the concentration is 100 micromolar. If we convert the units to a percentage, which is 0.001 to 0.002% weight per volume, very low concentration. We are sure it will not affect the sensory properties of the food. And also, we combine these low concentration of compounds with the UVA irradiation. The UVA intensity was 4.4 milliwatts per centimeter square. What is that intensity? This intensity is equivalent to the UVA component in the sunlight we get from the Earth's surface. So it's not the whole wavelength sunlight, it's just the UVA component we get from the sunlight. So we are sure the whole wavelength sunlight, the intensity would be much higher than this value. So we tested the E. coli and the Listeria, and we found that E. coli is more resistant to this approach, but with our two compounds still exert higher antimicrobial activity as compared to the rose bangle. And for the Listeria, every compound works very well. So the conclusion is that using the high throughput screen approach, we can identify compounds which have the high antimicrobial activity for both gram-positive and gram-negative bacteria. So some potential applications and some ongoing work in our lab. We are still working on the discovery of more food-grade compounds, both natural and synthetic compounds. And we can use this approach for the sanitation of food surface and the food contact surface. And also we can use it for the drinking waters disinfection, like the solid systems. And we can add these compounds to the juice of beverages. Sometimes these compounds are already in the juice of beverages. So we can turn on our UVA light, then bacteria can get inactivated. And also we can use this, utilize the sunlight and to treat the irrigation water. And also sometimes we can use this approach for the dental applications. So again, these compounds can also be immobilized on the food equipment surface, as well as the active packaging and the biomedical surface. So this is the first part I want to introduce today, and let's move to the second part. The second part I would like to talk about is the nature-inspired antimicrobial delivery system. So we know the food system is usually very complex because there are the presence of organic content and different surface structure of the food. So we really need some target delivery systems which can help us to deliver these food grade antimicrobial compounds. So in this case, we use the brewing byproduct, the yeast cells, and then we encapsulate the antimicrobial compounds into the yeast cells. So using a fast approach, which is the vacuum infusion, it only takes 5 to 30 seconds. Then we have the antimicrobial composite. So this is a versatile and a rapid approach for encapsulating a broad spectrum of antimicrobial compounds. And we got some patents and publications around this work. So as we said, it can encapsulate broad spectrum of antimicrobial compounds. So first of all, it can encapsulate N-haloamine polymers. As we know, N-haloamine polymers can bind chlorine to the polymer molecules. So we can have these molecules will have the antimicrobial activity. And also because the unique feature of N-haloamine polymers, these polymers can be recharged by the chlorine. So secondly, we can encapsulate hydrophobic bioactives like the curcumin. And then we can encapsulate essential oils like the lipophilic plant extracts. So recently we successfully encapsulated the Manuka essential oil into the yeast cells by this vacuum infusion approach, and we just submit our manuscript. And then we can also encapsulate metallic nanoparticles and other, as well as other nanomaterials. So today I just take one example. We are talking about the N-haloamine polymers. So these polymers can be charged by chlorine, because chlorine is the cheapest sanitizer and widely being used in the food industry. So the unique feature of our approach is that we are not modifying the particle surface. I know there are a lot of nanoparticles or microparticles studies there, but we are using the food grade ingredients, and also we are not modifying the particle surface. We are loading the antimicrobials inside the particles. That means we can get a very high concentration of antimicrobials. And secondly, these yeast cells have high affinity to bind diverse microbes. So we can get a target delivery of antimicrobials. And then because we are loading the active compounds inside the particles, so these particles have the control release property. And also these particles can protect the encapsulated antimicrobials from the harsh environment, like the organic content or the pH values, low pH values. We know the organic content is a big problem for the chlorine sanitizer. So the hypothesis is our particles can bind the microbes on the fresh produce or on the food contact surface. And once they bind, they can release the free chlorine to the wash water, and this free chlorine can kill the microbes, but they can also kill the microbes by direct contact between the yeast cells and the bacteria. So once they kill the bacteria, because the presence of particles, they can enhance the shear stress around the food surface. So they are not only kill the bacteria, they can also remove the dead bacteria from the food surface or the food contact surface. So that means we can enhance the antimicrobial activity, make the chlorine effective at low doses. So here's the basic process. We can catch the yeast microparticles, and then we load the antimicrobials inside the particles, and then we can expose our particles to 1% household bleach. Then this chlorine can bond to the particles. So some applications, if we use these particles for the sanitation of fresh produce, so we can get much low microbial population existing on the fresh produce surface as compared to the 50 ppm of free chlorine. And also we tested the chlorate and the perchlorate compound concentration remained on the lettuce surface after the washing process. So our particles doesn't increase the perchlorate and the chlorate concentration on the leaf surface. We can see the 50 ppm chlorine significantly increase the chlorate concentration on the fresh produce surface. And also we can use this approach to prevent the food spoilage. So we have tomato, and we inoculate the tomato with botrytis strains, and then if we spray with the 100 ppm of free chlorine, and after 4 to 5 days, the tomato still gets mold. But if we use even low concentration, 50 ppm of our chlorine our particles, these tomatoes didn't get any mold after 4 to 5 days incubation. And then we can use this approach for sanitation of biofilms on the food contact surface. So in this case, we grow the 4 days E. coli biofilm on the plastic surface, and our approach can get 7 log reduction in 20 minutes. And if we use the conventional chlorine-based sanitizer, it only get 1 to 2 log reduction. And our recent work is to immobilize these microcarriers, the yeast cells, onto the plastic surface by the chemical conjugation approach. So we can get these antimicrobial coatings on the plastic surface, and we can chlorinate the material, and then this chlorine can be charged on the material, and once they contact with the bacteria, bacteria can be killed, but the killing process will also consume some chlorine. But if we re-expose the plastic films to the 1% household bleach, and the chlorine can be recharged. So here is the photograph of the native plastics material and the modified plastic material, and there is no significant visual difference. And our material has a very good self-cleaning property. If we inoculate 6 log of bacteria on the surface, and we can get 5 log reduction in a very short period of time. So we can also use these particles for the edible coatings. So we immobilize, we coat the like the cleaning sponges or brushes with these particles, and we can get significant. We can prevent the cross-contamination between the fresh produce and the sponges or brushes. So some ongoing work, we are explore more bio-based targeted delivery systems, and also we are modifying the particle surface and to make them more specific to target bacteria. And yeah, so our lab is targeting on the bio-based approach. So we also working on some bacteriophage-based formulation. So we have the chemical approach to stabilize the phage against the UV light or other desiccation, some harsh environment. We are also developing some antimicrobial and anti-foreign materials for the food contact surface applications. So that's it from me, and I'm happy to take any questions. Thank you.
Thank you very much for that talk. We do have just one question for yourself, and that is, how effective is the treatment against bacterial and fungal spores?
At this moment, we just tried bacteria, and for bacteria, it works very well. So we usually will get like 5 log, 6 log reduction within 30 minutes treatment. Sometimes even shorter time, but we haven't tried the spores. For fungal, for fungi, we just do one experiment using the yeast cells with anhylamine polymer charged with chlorine. It can still work better than the conventional chlorine-based sanitizer, but of course the fungi would be more resistant than the bacteria. So, but we still can get like a 3 log reduction. Yeah.
Okay, thank you very much. Thanks for that presentation. For those of you whose questions we didn't get to answer, there will be answers that will be put up on the website, which is www.nzfssrc.org.nz. So I would like to thank all the presenters today for their excellent presentations, and just to remind everyone that the next webinar series will be on the 29th of September at 1:30, and it is entitled Tracking the Bad Bugs Faster. So thank you very much for your participation. Kath, I don't know whether you wanted to say a couple of closing words.
No, just thanks for coming, everybody, and we look forward to seeing you again, and a big thanks to the presenters. Great talks, very interesting, and thanks to you, Sue.
Thank you.
Great, see you, everybody.